Hi! In this video, I hope to walk through the steps of how to deduce a chemical structure using analysis of mass spectrometry, infrared spectrometry, and proton and carbon NMRs. The specific molecule that we are trying to figure out its structure is C3H5Br02. Using crucial information from the aforementioned data sets, we will be able to come up with a plausible structure for this molecule. I'm going to start off by looking at the mass spec graph. In the lab, a molecule is bombarded with electrons, which causes it to fragment off into charged particles. These charged particles are then grouped according to mass and abundancy, which is what are represented by the X and Y axes respectively. That's why a mass spec graph is a great place to start, as it gives you an idea of what the structural formula is, as well as what particles are most abundant. So now I'm going to go ahead and figure out the degrees of freedom. Remember, when determining degrees of freedom, you treat oxygens as if they do not exist, and halides as if they are an extra hydrogen. This is because oxygen is capable of making 2 bonds, and a halide is only capable of making 1 bond, so it acts as if it's a hydrogen. So let's go ahead and take the formula 2n plus 2 to represent the number of hydrogens we should have if there were no degrees of unsaturation. I've gone ahead and done the simple math, and we should have 8 hydrogens. However, we only have 5 plus the bromide, which makes 6, ignoring the oxygen. So we only have 6 hydrogens. So if we go ahead and divide this, or subtract this, we get 2, and then divide by 2, we have 1 degree of unsaturation, which means either we have 1 ring or 1 double bond, something along those lines. Also, if we wanted to go ahead and start digging into this mass spec, we can look at what possible fractions of particles could have broken off, say, between these 2 points. So the difference here is 17, and something that comes to mind right away would be an oxygen, which has 16, plus a hydrogen. So an alcohol group could have broken off right here. Moving forward, we'll see that this difference between 135 and 107 is 28, and something that could have broken off would be a CH2CH2 group, or a C double bonded to an O. So as you can tell, we're already running into some problems, as mass spec involves a lot of guessing, and you're not exactly sure of what exactly broke off, just the weight of what broke off. So we'll move on to IR to hopefully deduce some more information. So here's the IR spectrum graph. In the lab, an infrared light is used to interfere with bonds between certain molecules, which causes them to essentially dance, as Professor Zaba would say. So the motion and the vibrations of the stretching of the bonds are tracked on this graph, which is where this comes from. Over time, certain bonds between molecules have been recognized, and the shape of them has been recorded. So I referred to a data table from UC Davis that tracked the 5 zones of an IR spectrum. So one of the zones that is really recognizable is the alcohol zone, which happens to be very apparent right in this region. And this supports our idea that there was an alcohol group that had broken off in the MS spectrum. Moving forward in this 1700 region, we see the presence of what seems to be a C double bonded to an O. So this gives us another clue of what our structural formula looks like. As a reminder, the IR spectrum is really useful for determining what kinds of functional groups or what is present, but it doesn't necessarily tell you how much, which is what the MS was used for. Lastly, there's also an indication of a CH-sp2 bond right here in this area, which we will mark in green. So far, we can confirm that some of the fragments that we detected from the MS spectrum are in fact present, as you can see an alcohol group, CH bond, and a C double bonded to an O, which could be indicative of, say, a ketone. As we move forward to the proton NMR, hopefully we will be able to begin guessing what kind of structure it is. So here we'll start with the proton NMR graph. As you can see, there are 3 peaks, with 2 of the peaks being very, very strong. Using calculus and integration, the area under the peaks has been calculated for us. As you can tell based on the integration, there are 2 groups of 2 hydrogens together and then 1 hydrogen standing alone. In order to be able to visualize the number of peaks or the splitting that's happened for this NMR, I've gone ahead and zoomed in. As you can tell, both of these groupings have been split into 3 peaks. Using the n+1 rule, we can tell how many neighboring hydrogens each respective grouping can see, quote unquote. So because there are 3 peaks and there's an n+1 rule, that means that there are 2 neighboring hydrogens that both of these peaks can visualize. So I'm going to go ahead and zoom back out and label what we just discovered with the peaks. So here, this sees 2 neighboring ones, and this sees 2 neighboring ones, as they both have 3 peaks respectively. If we go back to our hypothesized molecule, we can start using deductive reasoning to eliminate some of our choices. So to the side here, I'm going to go ahead and draw out 3 potential molecular structures to our C3H5BrO2 molecule. So as you can notice, I've just shifted around where the halide is or where the ketone is. So while there are very slight changes, the proton NMR can actually reveal which structure is correct. Ultimately, this splitting and the ability to find the area under the curve to find the grouping of the hydrogens is very, very useful. Also, you can notice in all the 3 structures, I try to incorporate all the clues that we figured out from the previous MS and IR graphs. So I've included alcohol groups and ketone groups in all of them. However, right off the bat, you can tell that the first one already does not have enough hydrogen, and also the grouping is off, as this group right here would be 3 protons. So this is not correct. Looking to consider choices 2 and 3, we can focus on the splitting. So if the third structure were in fact the right one, then this carbon right here would not have any splitting, as it does not have any neighboring hydrogens. So we should have seen just a single peak. However, that's not the case, as we see 2 signals that are split into 3 peaks each. So that's not it. So now let's go ahead and check if our second one fits all the criteria. So you can see that at both of these carbons, they would have 2 neighboring hydrogens each, which fulfills our curve of having 3 peaks for both of those. Also, this one hydrogen on its own has very broad exchanges, and they are grouped with 2 hydrogens each. So it seems like this is their correct structure. We can now move on to the carbon NMR to see if that can confirm our guessed structure. As you'll notice, a carbon NMR is not as useful as a proton NMR, as it doesn't allow you to see splitting of peaks or integrate the area under the curve. So it doesn't reveal that much, but after consulting an online data table of known carbon shifts, I can confirm that our structure is correct. Here we can visualize a peak at 178.6, which is indicative of a aldehyde, or a carbon double bonded to an oxygen, which should fall somewhere between 160 to 180. Next, we see this peak at 24.3, which could indicate an CH2, which is supposed to fall in a chemical shift somewhere between 16 to 25. Lastly, we see this peak here at 38.6, which indicates a carbon bonded to a halide, so let's just say Br, which should fall somewhere between 30 to 60. Once again, we can tell that all of our graphs and data that was provided confirm that the structure we have guessed is in fact the correct one. Here is what our molecule would look like in 3D. Hopefully this video has helped you learn how to assess the MS, IR, proton NMR, and carbon NMR to deduce this molecular formula and structure for a given molecule. Thanks for watching!